A drug discovered in the soil of Easter Island extends lifespan in every animal species ever tested. Mice, flies, worms, dogs. The results replicate across every phylum researchers have tried. Most people have never heard of it because it was approved for a different purpose, and the patent that once protected it expired before anyone with financial incentive could reposition it for aging. By the end of this video, you will understand a biological pathway that controls how fast every cell in your body ages, and why the drug that modulates it sits in research journals instead of clinical practice. But the most disturbing part is not what the drug does. It is what the research found when they looked at who was already taking it and what happened to them. In 1972, a Canadian research team collected soil samples from Rapa Nui, Easter Island. Inside those samples was a bacterium producing a compound unlike anything in the existing pharmacological library. They named it Rapamycin. By 1999, the FDA approved it, not for aging, but for preventing organ rejection after transplant surgery. That approval changed nothing about what the drug was actually doing inside cells. In 2009, a landmark study from the National Institute on Aging tested rapamycin in mice, beginning treatment at an age equivalent to 60 human years. Life expectancy in those mice increased by 14% in females and 9% in males. The drug extended life even when started in late middle age. That finding has been replicated in every mammalian model since. Right now, inside your body, a protein complex called mTOR is making decisions about whether your cells repair themselves or push toward growth. Rapamycin is the only known compound that reliably shifts that decision. A 2020 study at the University of Washington found that intermittent rapamycin dosing in middle-aged dogs improved cardiac function and reduced biological age markers in a randomized controlled trial in companion animals with owner consent. But the mechanism behind those findings is not what the transplant literature describes. And what it reveals about why your cells age at all is something medicine has not built a framework to address. Inside every cell, mTOR, mechanistic target of rapamycin, functions as a resource allocation switch. When mTOR is active, the cell builds. It synthesizes proteins, it divides, it grows. When mTOR is suppressed, the cell shifts into a different mode entirely. It stops building and starts cleaning. It runs a process called autophagy, the systematic digestion of damaged cellular components that would otherwise accumulate over decades. Think of mTOR as a foreman who never lets the crew stop working. Autophagy is the maintenance shift. It only runs when the foreman steps away. In a young body, mTOR cycles naturally, active periods followed by quiet periods that allow cellular cleanup. With age, that cycling breaks down. mTOR stays active longer. The maintenance shift runs less often. Cellular debris accumulates. This is not metaphor. Autophagic decline is measurable in tissue samples from humans in their 30s. Rapamycin suppresses mTOR selectively. It creates a pharmacological maintenance window. The cell runs the cleanup it would have run in youth, without requiring the caloric restriction or fasting that would otherwise trigger the same response. The drug does not add anything. It removes the signal that tells your cells to stop repairing themselves. And here is where the structure of medicine becomes the story. Because the reason rapamycin is not being tested in healthy adults for longevity has nothing to do with the science. It has to do with a regulatory category that does not exist. And the consequences of that absence are not abstract. The FDA does not recognize aging as a disease. It is classified as a natural process. That classification means no pharmaceutical company can run a phase 3 trial with slowing biological aging as the primary endpoint, because there is no approved disease indication to approve a drug against. The regulatory category does not exist, so the trial cannot be designed. This is the structure. A drug approval system built for acute conditions, applied to a chronic, universal, measurable biological process, and rendering it invisible by refusing to name it. The concrete example is this. Rapamycin is currently prescribed off-label by a small network of longevity physicians, mostly in private practice, mostly cash pay, mostly to patients who already know to ask for it. That network has no standardized protocol and no regulatory oversight. Your doctor has almost certainly never discussed rapamycin with you. Not because the evidence is absent. The 2009 NIA study is one of the most replicated findings in geroscience, but because prescribing it for aging would constitute off-label use with no insurance pathway and significant liability exposure. A 2023 survey of longevity physicians found that rapamycin was the most commonly prescribed off-label intervention among practitioners explicitly focused on biological age. It is not available through any standard insurance plan for that purpose. It costs between $30 and $60 a month out of pocket. The intervention costs less than most monthly gym memberships. It is not discussed in any standard annual physical anywhere in the world. What makes this structural rather than incidental is the incentive geometry. Rapamycin's patent expired in 2009. No company profits from prescribing the generic. The research pipeline for off-patent molecules in aging runs almost entirely on public grant funding, which is a fraction of what pharmaceutical marketing budgets spend in a single quarter. But what the clinical evidence, the human evidence, actually shows about rapamycin's effects is more specific than the animal data suggests. And that specificity changes what is possible to do right now. 3 findings from human data are worth separating from the animal literature. They are not equivalent to the mouse lifespan studies. They are more useful. First, a 2014 study published in Science Translational Medicine found that low-dose rapamycin improved immune response to influenza vaccine in adults over 65 by up to 20%. The mechanism was mTOR suppression, restoring immune cell function that had declined with age. Second, a 2019 paper in the journal Aging Cell found that rapamycin reduced markers of cellular senescence, the accumulation of damaged cells that no longer divide but continue to emit inflammatory signals, in human tissue samples taken from older adults. Senescent cells are the biological equivalent of a broken machine that cannot be removed but keeps disrupting the factory floor. Rapamycin does not remove them. It reduces how aggressively they disrupt what surrounds them. Third, the dosing protocol matters more than the dose. Continuous daily rapamycin, the transplant dose, suppresses immune function dangerously. Intermittent dosing at a fraction of that level appears to produce the mTOR suppression benefit without the immunosuppressive risk. That distinction is what the off-label longevity community has built its protocols around. Your biology is not fixed at its current state of cellular maintenance. The mTOR pathway responds to signal. The question is whether the signal it is currently receiving is the one you would choose to send. Every year that passes without addressing autophagic decline is a year of compounding cellular debt that the research suggests becomes harder, not impossible, but harder to reverse. In 1972, a soil sample from a remote Pacific island contained a compound that would eventually extend life in every animal model science applied it to. That was 52 years ago. The scientific community knows what rapamycin does to mTOR. It knows what mTOR does to aging. It knows the dosing distinction between immunosuppression and longevity effect. That knowledge exists in published, peer-reviewed form, available to any researcher who searches for it. What does not exist is a system designed to move that knowledge from the journal to the prescription pad. The TAME trial, the first FDA-approved clinical trial targeting aging as a process using metformin rather than rapamycin, launched in 2023. It will produce results in approximately 6 years. Rapamycin has no equivalent trial currently funded at scale. The drug that works in every animal tested is waiting for a regulatory category that does not yet exist. And the cells making decisions about your repair cycle right now are not waiting with it.